take a genetically optimized, fast-growing broiler chicken and you just place it at 11,000 feet in the Trans-Himalayan desert, its heart will literally pump itself to death trying to find oxygen.
Yeah, it's a completely fatal biological cascade.
Right. But today we're doing a deep dive into a really incredible study that actually stopped this cascade. And they did it using something honestly completely unexpected, which is agricultural waste from local apricot trees.
Which is wild, right? I mean, the environment we're looking at here is the Leh-Ladakh region. And for those of you who aren't familiar, it is the absolute definition of an extreme survival test.
Oh, for sure.
We're talking about an environment defined by hypobaric hypoxia.
Meaning the atmospheric pressure is just so low that the actual density of oxygen molecules in the air is, well, it's dangerously sparse.
Exactly. And then you add in this intense ultraviolet radiation tearing through the thin atmosphere, plus incredibly low humidity, scarce vegetation, and these wild temperature swings.
Yeah, the temperature swings are crazy.
I mean, it can hit a scorching plus 35 degrees Celsius in the summer, and then just plummets all the way down to minus 35 degrees in the winter.
So if you're a farmer trying to raise livestock in those conditions, you are just fighting a constant battle against geography.
Oh, absolutely.
And the sources we're unpacking today detail how these environmental extremes place this enormous physiological stress on broiler chickens. And the result is metabolic disturbances that weaken their immunity, stunt their growth, and lead to these massive mortality rates.
Which obviously devastates the profitability for the local poultry farmers.
Exactly. But what I love about this study is that the researchers didn't try to solve this problem with some expensive synthetic drug or massive climate-controlled facility.
Right. They looked locally. They looked at this local botanical survivor, Prunus armeniaca, the apricot tree.
The apricot tree.
Yeah. And they hypothesized that the seeds of this plant might contain the exact biological armor these birds need to survive the hypoxia.
Okay, let's unpack this a bit. To understand why a local seed could be the solution, we first need to really look at the victims here. Because, I mean, it seems kind of counterintuitive that a bird would struggle at high altitudes, right?
Yeah, you'd think birds belong in the sky.
Exactly. You look at something like the bar-headed goose, which naturally migrates directly over the Himalayas without an issue. So why does a broiler chicken fail so spectacularly just standing on the ground at 3,500 meters?
Well, the vulnerability really lies in their genetic selection.
Right.
Wild high-altitude birds have evolved over millions of years to optimize oxygen binding in their hemoglobin. They maximize their lung capacity. But modern broiler chickens, they have been selectively bred over the last few decades for one highly specific trait.
Maximum muscle accumulation.
Yes, in the shortest possible time. They are biologically programmed to just grow massive amounts of tissue incredibly fast.
And building that much tissue requires an immense amount of cellular energy.
Exactly. And generating that energy requires oxygen. Oxygen is it's the terminal electron acceptor in cellular respiration.
It's the critical molecule that lets a cell cleanly process glucose into usable energy.
Right. So because these broilers grow so incredibly fast, their metabolic rate is just exceptionally high, which means their oxygen demand is staggering.
So let's visualize this for you listening. Think of the chicken's cellular metabolism as a high-performance combustion engine.
Oh, that's a good way to look at it.
Right. So in a normal environment, like down at sea level, the engine takes in fuel, which is glucose from their feed, and mixes it with a heavy intake of oxygen. And this results in clean, efficient combustion that powers the rapid growth of the bird.
But the physiological mismatch happens when you transport that high-performance engine up to Leh-Ladakh.
Because the broiler's genetics are still demanding maximum fuel combustion.
Exactly. But the hypobaric hypoxia means the oxygen intake is severely restricted. So the cellular engine is running far too rich on fuel and just desperately lean on oxygen.
Right, so the combustion process breaks down. Like in a car engine, you'd get misfires and these rogue sparks bouncing around the engine block.
Yeah.
So what's actually happening inside the chicken's mitochondria when that oxygen deficit hits?
Well, the electron transport chain inside the mitochondria begins to stall out because there just isn't enough oxygen to safely accept the electrons at the end of the energy production cycle.
Okay.
So those electrons literally leak out into the surrounding cellular fluid. And they prematurely bind with whatever sparse oxygen is actually available, which creates these highly unstable molecules called free radicals.
Or reactive oxygen species, right? ROS.
Exactly, ROS.
So those leaked electrons are the rogue sparks from our engine analogy.
That is exactly what they are. And just as rogue sparks would eventually, melt an engine block from the inside out, these free radicals cause severe internal destruction.
Because a free radical is essentially just a molecule missing an electron, making it violently reactive.
Right, it's desperate. It will aggressively tear an electron away from the nearest stable structure just to stabilize itself.
Yeah.
And if they aren't neutralized, they trigger a state of severe oxidative stress.
Wow.
Yeah, they damage DNA, they warp vital cellular proteins, and they trigger this massive systemic inflammatory response.
And the sources single out one specific super deadly condition that this oxidative stress causes in these high-altitude flocks, which is ascites.
Yes, ascites is terrible.
Can you walk us through how a cellular spark leads to a fatal fluid buildup in the bird's abdomen?
Yeah, it's this really devastating chain reaction. So the hypoxia and the oxidative stress basically damage the endothelial cells lining the blood vessels in the lungs.
Okay.
And that causes those vessels to constrict. So the chicken's heart realizes the body's starving for oxygen, right? So it starts pumping harder, just trying to force more blood through those narrow, damaged pulmonary vessels.
Which causes severe pulmonary hypertension.
Exactly. The right ventricle of the heart becomes dangerously enlarged from all that immense pressure, and eventually the valve just fails.
Oh, wow.
The right side of the heart gives out, and blood plasma literally begins to leak directly into the abdominal cavity.
The heart literally just works itself to failure. I mean, it's a grim reality for these farmers. You've got a bird that is biologically programmed to run a marathon, placed in an environment where it cannot catch its breath.
Right.
And that results in oxidative stress that just destroys the cardiovascular system.
Which, thankfully, brings the researchers to the apricot tree.
Yes. Here's where it gets really interesting.
Because think about it. These trans-Himalayan plants have sat in this brutal, high UV, freezing, hypoxic desert for literally millennia.
Right. They can't exactly run inside to avoid the elements.
No, they can't. So they had to evolve these intense internal biochemical defenses to survive the exact same oxidative stress that is killing the chickens.
But, while the sweet, fleshy fruit of the apricot is widely consumed and exported, the researchers completely ignored the fruit.
Yeah, they didn't care about the fruit at all. They went straight for the waste product, the kernel or the seed locked inside that hard pit.
Because when the lab analysis was done on these kernels, they proved to be total phytochemical powerhouses.
Oh, absolutely. They are exceptionally rich in biologically active compounds. We're talking polyphenols, flavonoids, carotenoids, and a bunch of essential vitamins and fatty acids.
Now, we hear terms like polyphenols and flavonoids tossed around constantly in, human nutrition and supplement marketing.
Oh, totally. Superfoods, right?
Exactly. But functionally, what are these molecules actually doing that makes them so valuable in this extreme environment?
Well, at a molecular level, these compounds act as the ultimate biological grounding wires for those rogue sparks we talked about.
Oh, I like that grounding wire.
Yeah. Polyphenols like catechins, neochlorogenic acid, and caffeic acid, they have these unique chemical structures that allow them to easily donate an electron to a free radical without actually becoming unstable themselves.
Ah, so they neutralize the reactive oxygen species before they can rip apart the chicken's cellular membranes.
Exactly. It's why these natural phytochemicals have been valued in local folk medicine for generations. They have incredible anti-inflammatory, hepatoprotective, and cardioprotective properties.
Okay, but wait. If you look at an actual apricot pit, it is essentially a piece of hardened wood.
Yeah, it's super tough.
Right. And the seed inside is fibrous and hard. You can't simply dump a bucket of raw apricot kernels into a chicken trough and expect these microscopic polyphenols to just, magically absorb into the bird's bloodstream.
No, definitely not.
So how did the scientists physically extract this biological armor?
You're absolutely right. The raw form is totally inaccessible to a chicken's digestive tract. So the researchers used this really meticulous extraction process.
Okay.
First, they sourced dried apricot kernels from the local flea market and ground them up into a highly uniform, really fine powder. Then they subjected that powder to what's known as a sock slit extraction using distilled water.
Okay. For those of us who haven't been in a chemistry lab recently, how exactly does a sock slit extractor work?
Okay, imagine a highly sophisticated coffee percolator.
Oh, perfect. I know coffee.
Right. So you place the raw powder in a porous thimble inside a glass chamber, and then distilled water is heated in a flask right below it.
Okay.
The water vapor rises, condenses, and slowly drips down through the apricot powder, dissolving those water-soluble polyphenols and flavonoids.
Wow.
And once the chamber fills up, a siphon mechanism flushes that polyphenol-rich water back down into the heating flask. And this cycle just runs continuously over and over for hours.
Just repeatedly washing the powder with fresh solvent.
Exactly, pulling out every available bioactive compound from that really tough plant matrix.
Okay, but after they pull all those compounds into the water, how do they stabilize it? Because I imagine you can't just ship vats of apricot tea to the farms, right?
No, that would spoil instantly. So they used a rotary evaporator to gently pull the water away under vacuum pressure, which leaves behind this highly concentrated extract.
Got it.
But then, to make sure the delicate antioxidant compounds weren't destroyed by heat, they actually freeze-dried the extract at ultra-low temperatures, like between -88 and -90 degrees Celsius.
Wow, that is cold.
Yeah, and that locked the bioactive molecules into a really stable dry powder.
But moving from a localized lab setup to a live flock is a massive biological leap. I mean, they didn't just start feeding this powder to the chickens on day one, did they?
No, they had to be careful. They conducted preliminary in vitro investigations first.
Okay, so petri dish stuff.
Exactly. They cultured chicken peripheral blood lymphocytes, which are key immune cells, in a petri dish.
Yeah.
They needed to make sure the extract wasn't cytotoxic. And actually, they found it stimulated the immune cells to proliferate.
Oh, nice.
But the true test of its protective power was the hydrogen peroxide challenge.
Right, because hydrogen peroxide induces massive oxidative damage at the cellular level.
Yep.
It chemically mimics the exact kind of free radical storm the chickens experience at 11,000 feet.
Precisely. Yeah. So when they intentionally exposed the immune cells to the hydrogen peroxide, the cells naturally began to degrade. But when they introduced the apricot seed extract alongside the hydrogen peroxide,
Extract shielded them.
it completely shielded the lymphocytes. The polyphenols intercepted the oxidative damage, which confirmed the potent cytoprotective properties of the extract before a single live animal was ever involved.
So the petri dish proved the concept. But, I mean, protecting a single cell layer in a controlled, warm incubator is entirely different from protecting the complex physiology of a living, breathing, rapidly growing broiler chicken up at 3,500 meters.
Oh, completely different.
So how did they structure the actual in vivo trial?
Well, they set up a rigorous 42-day trial, which represents the standard growth cycle for a commercial broiler before it reaches market weight.
Yeah.
It took 105 one-day-old broiler chicks and randomly assigned them to 7 different experimental groups.
So they were basically actively searching for the therapeutic window because giving the birds too little of the extract wouldn't provide enough protection, but feeding them too much could cause adverse metabolic reactions or, just be way too expensive for the farmer to sustain.
Exactly. To find that precise balance, one group was designated as the control. These birds received just a standard basal diet and plain water, leaving them totally exposed to the high-altitude stress without any added defense.
Okay. And the other 6?
The remaining 6 treatment groups received the apricot seed extract dissolved directly into their daily drinking water at escalating concentrations.
Ramping it up.
Yep. 100, 150, 200, 300, 400, and all the way up to 800 milligrams per kilogram of body weight.
Wow, quite a range. So during those 42 days, what specific metrics were they watching? Because obviously they were looking at weight, but it sounds like they went much deeper into the underlying physiology.
Oh, the monitoring was incredibly comprehensive. They tracked daily growth performance and feed utilization, which, tells us how efficiently the birds are converting their food into muscle rather than just burning it off as stress heat.
Here comes this.
They meticulously recorded mortality rates. And most importantly, they drew blood samples on day 0, day 21, and day 42.
To look at what?
To analyze the circulating antioxidant levels, liver enzymes, and inflammatory markers, they really wanted to document the real-time biochemical changes occurring inside the birds.
All right, so the 42 days conclude. The birds are weighed, the blood is spun down and analyzed, and the results from this trans-Himalayan trial are finalized. Let's look at the actual growth numbers. What happened?
The overarching finding was deeply significant. Every single group that received the apricot extract, regardless of the specific dose, exhibited a significantly greater final body weight than the untreated control group.
Every single one. That's amazing. But the data reveals a very clear, Goldilocks dose, right?
It does.
The 200 milligram per kilogram group, which they labeled as the T3 group, completely outpaced the rest.
Yeah, T3 was the sweet spot.
Those specific birds reached an average weight of 450.14 grams, while the control group, which received no biological armor, struggled to reach even 348.53 grams.
Which is a massive discrepancy in growth efficiency.
Absolutely massive.
Yeah, because the birds in the T3 group weren't wasting their daily energy reserves fighting off hypobaric stress and systemic inflammation. Their cellular engines were actually running cleanly, so the energy from their feed was actually being directed toward muscle synthesis.
The weight gain is impressive, for sure, but I think the mortality data is the true centerpiece of this whole study.
Oh, without a doubt.
The control group, left to fend for itself at altitude, suffered a 20% mortality rate.
1 out of every 5 birds died.
Yeah, primarily succumbing to ascites and that cardiovascular failure we talked about. But when you look at the T3 group receiving the 200 milligram dose, what was the mortality rate?
It plummeted to absolute zero.
Wait, really?
Zero. Not a single bird in the 200 milligram treatment group died during the entire trial.
Wow. The survival of the entire flock is an incredible outcome. But, to prove it wasn't just some crazy anomaly, we have to look at the blood biochemistry.
Right, the under-the-hood numbers.
Exactly. How did the researchers chemically prove that the apricot extract was actually responsible for stopping the cardiovascular failure? Let's talk about the MDA levels.
Okay, so MDA stands for malondialdehyde.
Malondialdehyde. Okay.
And it is a critical biomarker for a destructive process called lipid peroxidation.
What is that?
Well, the membranes that encapsulate every single cell in the body are primarily composed of lipids or fats.
Okay, sure.
So when those rogue free radicals we discussed earlier bounce around looking for an electron, they frequently tear that electron straight away from the lipid bilayer of the cell membrane.
So they literally rip the structural walls of the cell apart.
Exactly. And MDA is the metabolic debris left behind by that microscopic destruction. So high MDA in the blood means widespread cellular damage is actively occurring.
Like chemical rusting.
Yes, perfect analogy. Rusting. And in the untreated control group, these circulating MDA levels were dangerously elevated. The hypoxia was just causing massive lipid peroxidation.
But in the treatment groups?
In the treatment groups, and particularly that T3 group, the MDA levels were drastically suppressed. The apricot polyphenols were successfully intercepting the free radicals before they could even strike the cell membranes.
So the chemical rusting of the cellular walls was totally halted.
And the blood tests also measured TAC, or total antioxidant capacity.
And I'm guessing that went up.
Oh yeah. As you would logically expect, the extract-treated chickens demonstrated a vastly superior overall capacity to scavenge and neutralize oxidative threats in their bloodstream compared to the control group.
But it goes even deeper than just neutralizing the free radicals, though.
It does.
Because the data shows the extract actively modulated the immune system's panic response. The researchers looked at specific inflammatory cytokines, which are the chemical messengers the immune system uses to communicate.
Right. And they specifically focused on interleukin-6, or IL-6.
Okay, what does IL-6 do?
It is a really potent pro-inflammatory cytokine. When the body detects widespread oxidative damage, it floods the system with IL-6, which triggers this massive inflammatory cascade.
Oh, which can often cause more collateral damage than the initial stressor itself.
Exactly. But in the treated birds, the levels of IL-6 dropped significantly.
Wow.
Yeah, the apricot compounds were effectively signaling the immune system to stand down and just stop overreacting to the environmental stress.
But here's the really brilliant nuance in the data, I think. It didn't just blindly shut off the immune system.
No, not at all.
Because while the panic-inducing IL-6 went down, the levels of interleukin-2, or IL-2, actually increased.
And that right there is the hallmark of true immunomodulation rather than mere immunosuppression.
Right.
IL-2 is a cytokine that promotes the proliferation of T cells, which are the specialized cells that hunt down and destroy actual pathogens and infections.
So the extract lowered the destructive systemic inflammation while simultaneously sharpening the bird's targeted cellular immunity.
Exactly. And those protective effects even extended to the hepatic system. The sources detail how they measured AST and ALT.
Liver enzymes, right?
Yep. These are liver enzymes that normally reside safely inside healthy liver cells. But when the liver is damaged by oxidative stress or toxins, those cells rupture and the enzymes leak out into the bloodstream.
So what did the data show there?
Well, the untreated birds showed elevated AST and ALT, which indicates the high-altitude stress was seriously degrading their liver function.
Right.
But the groups receiving the 200 milligram apricot extract, they showed markedly lower levels of these enzymes in their blood, proving that the phytochemical shield was keeping the liver architecture completely intact.
So when you zoom out and synthesize all of these distinct biological victories, I mean, the interception of free radicals, the prevention of lipid peroxidation, the calming of systemic inflammation, the enhancement of T-cell immunity, and the protection of the liver, it just paints a really clear picture.
It does.
The molecular mechanics directly dictate the outward results. You get a flock with zero mortality that utilizes its feed with maximum efficiency.
And the researchers naturally took this a step further and ran the economic calculus for the farmers.
Oh, I bet they love that.
Oh, yeah. They factored in the baseline cost of raising the chicks, the cost of the feed, and that minor processing expense required to run the soxylate extraction on the apricot seeds.
Right, because it's not totally free to make the extract.
Exactly. But because the T3 group experienced zero mortality and achieved a significantly higher market weight, the net economic return for the farmer was vastly superior to the untreated flock.
It fundamentally changes the viability of agriculture in the Trans-Himalayan region. The study proves this isn't just, merely an interesting academic observation in a petri dish.
No, it is a highly viable, deeply cost-effective, phytogenic feed additive.
And it achieves all of this by utilizing a natural byproduct that was previously considered agricultural waste.
Right. The apricot trees cultivated in Ladakh had already solved the problem of oxidative stress through millennia of evolution. Human science simply learned how to extract that evolutionary knowledge and transfer it to a vulnerable species.
It's just so cool. To bring this full circle for you listening, we started by looking at an incredibly fragile biological system. A modern, fast-growing broiler chicken whose genetics demand a massive oxygen supply, placed in a cold desert that totally denies them that oxygen.
Yeah.
We saw how that mismatch causes the cellular engine to sputter, leaking those free radicals that tear apart the cardiovascular system and cause fatal fluid buildup. But by identifying the chemical defenses within the discarded kernel of the local apricot tree and determining that precise 200 milligram therapeutic dose, researchers provided these animals with a highly sophisticated biological armor.
Which leaves us with a really compelling broader implication to consider.
Oh, what's that?
Well, if the naturally evolved phytochemicals from a high-altitude plant possess such complex, potent defenses that they can completely rewrite the survivability, cardiovascular integrity, and immune function of a rapidly growing animal facing severe environmental trauma,
Yeah.
what other powerful biological armors exist out there?
Oh man, that is a profound thought. The natural world acts as an endless open-source library of survival strategies. If an overlooked apricot seed can halt heart failure at 11,000 feet, what undiscovered molecular tools are hiding in, like the root system of an alpine shrub or the bark of a desert tree, just waiting to be extracted and applied to modern medicine or global agriculture?
There's so much left to discover.
There really is. Thank you for joining us on this deep dive into the resilient mechanisms of the natural world. Keep asking questions, keep looking closer at the environments around you, and remember that sometimes the most sophisticated scientific solutions are hiding right inside the waste products we throw away.